Welcome. This is going to be our first video for Principles of Biology Lab. I'm actually in the Principles of Biology Lab here at ASUBB, all right? And our first lab exercise, all right, or the first topic, all right, for the lab is basically concerns the use of something referred to as the compound light microscope, all right? So again, basically once you're basically sitting at your lab bench right here, normally there's going to be a cabinet. It's either off to the right or off to the left, all right, that basically contains a piece of machinery referred to as the compound light microscope, all right? So the first thing you need to do is go ahead and pull that cabinet open and pull your compound light microscope out. Notice it's actually covered with a plastic microscope protection device, all right? What you're going to do is actually remove this and actually stick it back into the cabinet and actually close it off, all right? Now, when we're done, we'll actually stick this back on, all right, and actually put it away. So this instrument right here, which you may be using quite frequently, all right, not only in this course, but other more advanced courses, is called the compound light microscope, all right? So before I start talking about how we use this and the different parts of it and the different exercises that I've got planned for today, what is compound, all right? So compound is kind of like opposite what simple is. And so when I have a simple microscope, it's only composed of one lens, all right? An example is just a magnifying glass. A magnifying glass is only composed of one lens. But when I say the word compound, that basically means two or more, all right? So here, we're actually focusing on our specimen using two different lenses, all right? One lens, which we'll talk about just a little bit later, is called the ocular lens, all right? And typically the ocular lens is basically 10 times. What does that mean? So if I'm only looking at my specimen using the ocular lens, what I'm going to do is I'm actually going to magnify that specimen or that object 10 times, all right? Then the other type of lens, all right, because it's compound, is referred to as the objective lenses. And note, we've got at least 4 different objective lenses that are basically found on this compound light microscope. And so we can actually decrease or increase our magnification, all right? And notice again, each one has different types of magnification, 4 times, 10 times, 40 times, and 100 times. And we'll talk about that a little bit later on. Now, the second thing I want to point out, this is a compound light microscope, all right? So anytime you're viewing something, all right, you need an illumination source, all right? And for us, it's going to be a visible light, all right? The visible light actually comes down here from the light source from a light bulb, all right? But notice again, not all microscopes actually use visible light to actually view the specimen. If you basically go to an electron microscope, they basically use some sort of electronic beams, all right, that basically have very short wavelengths, all right? And so what they do is they can actually increase the magnification and also resolution even more, all right? And so again, compound using 2 or more lenses, and then light is our illumination source, and that's actually what we're viewing. I do want to make a point. Most of these compound light microscopes cost anywhere from $600 to $2,000. I'm not sure how much these cost, but a pretty good estimate is about $1,000, all right? So treat this with care, all right? Treat this with care because not only do I use it and you use it, all right, but other students use it, all right? So this is a compound light microscope. So first of all, before we actually start to plug it in and start talking about all the parts and how to use it, all right, notice that each microscope, regardless of whether or not it's a magnifying glass, all right, or a compound light microscope or an electron microscope, all right, all have 2 different characteristics, magnification and resolution. Obviously, magnification kind of makes sense because obviously something that's so small that we can't basically see with our eye, we want to magnify it to actually look at it, all right? So magnification is one of the big ones. But one thing I want to point out, most beginning students think, oh, the magnification is the key thing. Well, the second one is just as important, if not more important than magnification. That refers to resolution, all right? Resolution is when 2 small objects are very close to one another, all right? We're either going to see them as one object or 2 separate objects. And again, resolution is going to be big because when we stare at an object or a specimen through a compound microscope, the people like saying, well, how come we just can't increase the magnification to 10,000 or 100,000 or 100 million, all right? Well, it depends on our light source, all right, because the light, all right, is actually going to determine the resolution. So anything smaller than zero point 0.2 microns, all right, anything smaller than zero point 0.2 microns is going to appear as one object. Anything that's larger than zero point 0.2 microns is going to appear as two objects, all right? So although we can magnify it, magnify it, magnify it, because we're using visible light, all right, as our light source, we're not actually increasing our resolution, all right? So that's why we use other types of microscopes like confocal microscopes, all right? Or an electron microscope, all right? Because again, we can actually increase the resolution. We can actually distinguish between two objects instead of being one object, all right? So again, magnification and resolution are two important properties or characteristics of all microscopes, all right? And typically with the compound light microscope, the maximum amount of magnification that we can actually get is probably about 1,000. We'll talk about that in a couple of minutes. So next thing we want to do is we want to talk about the parts of the microscope, all right? So notice again, we do have to plug this in. Usually the electrical cord or the power cord is normally wrapped around the microscope. And notice again, it's wrapped around this structure right here, which is called the stage. And I'll talk about why we actually do it. Take it out, and if you want to, you can actually plug it in. I'm not going to, well, I guess I can plug mine in. So hold on. So I've actually plugged it in. So this is a compound light microscope. And so the first thing I want to do is talk about the different parts of a compound light microscope, all right? So what we're going to do is start up here. This area right here, this whole area right here is called the body tube, all right? This right here is called the body tube, all right? But notice that the body tube, all right, has these parts right here. This is called the eyepiece, all right? So eyepiece, body tube, all right? But notice that inside the eyepieces, all right, we're going to have lenses, all right? These are referred to as the ocular lens, all right? So the ocular lens will basically have a 10-fold or 10 times magnification. So what it's going to do is basically going to magnify your specimen or your object that you're viewing 10 times, all right? So again, 10 times, all right? Notice again, what I like about these microscopes is this also is referred to as the binocular. In other words, you're basically going to look at it through two eyes, all right, through two eyes. And notice when you look at it, make sure your glasses are off, all right? You can actually adjust this depending on the lengths of your eyes, all right? And in one of these, all right, there's actually going to be a pointer, all right? So I can actually pull things out, look at that pointer and tell me what you are viewing or what you're seeing, all right? And so again, eyepiece with the ocular lens. This is referred to as the body tube, all right? Now this structure right here, my glasses back on. This structure right here is called the arm, all right? This structure right here is called the arm. Then right below here, you're going to see 4 of these lenses right here, all right? These are referred to as the objective lenses, all right? The objective lenses. And so let's talk about the objective lenses before we discuss more parts of the microscopes. Notice again, we have this one, which is basically colored in red. It has like a little band of red around it. This is referred to as the scanning objective, all right? The scanning objective has a magnification of 4 times, all right? Then I'm going to twist this portion of the microscope right here. This right here is called the revolving nose piece. So I can increase my magnification, I can decrease my magnification. So again, objective lens, objective lens, objective lens, and this portion right here is the revolving nose piece. And so again, I'm going to go from my scanning objective, which is in red, to this objective right here, which is called the low power objective, all right? The low power objective is yellow, all right? And again, its magnification is 10 times, all right? 10 times. If I want to increase my magnification more, all right, I'm going to go from the low power objective to something referred to as the high power objective, all right? The high power objective is actually in blue, and it actually has a magnification of 40 times, all right? 40 times. Now, this last objective, we're probably not going to use in this lab, but for those of you who will be taking a microbiology class, all right, or sometimes we may use it in anatomy and physiology, it's this objective right here, which is white, all right, has a white bar around it. This is referred to as the oil immersion objective or the oil immersion lens, all right? We actually have to use immersion oil, all right, to actually view it, all right? But one thing I want to point out is for a beginning student, if you don't know how to clean that immersion oil off, it'll basically ruin your oil immersion objective or your oil immersion lens, all right? And typically, other than maybe if we do like a lab where we're actually looking at bacteria, we probably will not use this, all right? So again, we'll probably use the high power, the low power, and the scanning, all right? So let's talk about some other portions of the microscope. This area right here that you actually put your microscope slide on, I'm actually going to grab a microscope slide and actually show you how I do this. So I'm going to go grab a microscope slide and I can actually put it here in these little clips, all right? These little clips are actually going to hold my microscope slide onto something referred to as the stage, all right? So this entire structure right here is called the stage, all right? Now, notice it's sometimes specifically referred to as a mechanical stage, all right? And I'm basically showing you how old I am because when I was a kid, when we basically used microscopes back in like junior high or middle school, all right, we didn't have a mechanical stage. We actually had to use our fingers, all right, to basically move the microscope slide around, which was very difficult, all right? Here, it's a lot better, all right, because again, below the stage or the mechanical stage, let me turn this around, we have these two knobs right here. One knob is actually close to the stage. It's actually a little bit broader. And this other one, which is actually further away from the stage, is actually a little bit more narrower. Notice what's going to happen. When I twist this knob right here, that's basically close to the stage, notice my stage moves forward and backwards. Forward and backwards, all right? So this is referred to as the front to back adjustment knob, all right? So front to back adjustment knob. Now notice if I twist this knob down here, this is furthest away from the stage, it's a little bit more narrower. Notice when I do this, you can actually see the stage, it's actually going from left to right, left to right. So this is the right to left or the left to right adjustment knob, all right? So again, front to back adjustment knob, and this is the right to left adjustment knob, all right? Now what I'm going to do is I'm going to turn my compound microscope around, and what I'm going to do is I'm going to show you this thing that's right underneath the stage or the mechanical stage. This thing right here is called the substage condenser, all right, the substage condenser. It actually has a lens in it, but the lens is not basically due to magnification of the specimen. What it's doing is it's basically taking the light from the light source and actually projecting it, all right, through our specimen or the object that we're actually looking at, all right? So again, this is the substage condenser, all right? But notice, again, if you look at this, there's going to be a little lever, all right? This little lever right here can go one, basically minimal to maximal, all right? What you probably want to do is have this lever somewhere in the middle. This is something referred to as the iris diaphragm lever, all right? And it basically limits the amount of light coming through your substage condenser. So notice you can basically twist it to one side and you're going to have no light coming through your specimen whatsoever. Oh, come on. And the other one, if you twist it to the other one, all right, you're going to have maximal light coming through your substage condenser, all right? And notice if it's maximal, you may actually blind yourself when you actually look through the microscope. So put it in the middle. You may have to adjust it a little bit later on, depending on how it's stained, the thickness of your specimen, whatever, all right? But usually it's in the middle. Then we have, again, the light source, all right? So there's actually a light bulb, all right, that's actually attached here. I do want to make a point. When I turn my microscope around, I have my power switch, all right, here. So notice that it basically it's towards this big zero right here. It's basically off. When I switch it on, there's like a little line right here. That means the power's on. You could actually see that my light bulb is actually on. Now, I don't require you to know this, but notice there's also a little knob right below the power switch, all right? This is sometimes referred to as the rheostat because it actually adjusts the current or the electrical current going to the light bulb. So notice when I twist it, all right, this is maximal. When I start to twist it, notice it's actually decreasing the current going to the light bulb so I can actually dim my light, all right? So one of the things I want to point out, which a lot of students basically get wrong, all right, is when they turn on the microscope thinking like, well, my light bulb is burned out. And again, a lot of times that does happen. The light bulb is burned out, and it's very easy to basically replace the light bulbs in here. But when you turn it on and you don't see any light, that doesn't always mean that your light bulb is burned out. Check the rheostat first, and sometimes it takes a couple of seconds. So if you look at this, I'm going to twist it up. It's still not on. It's still not on. It's still not on. And bam, it's on. All right. So again, make sure your rheostat is adjusted so that you can actually see the light. All right. If you turn your rheostat all the way to full length and you probably don't see a light, all right, that probably means, hey, your light bulb is burnt out. All right. So again, power switch, rheostat. Obviously, this right here, this is the electrical cord, all right? One other thing I want to point out, a couple other things I want to point out are these two knobs right here. Notice you're going to have a knob that's a little bit thicker. It's actually going to be close to the microscope. And then you're going to have a knob that's a little bit more narrower, and it's actually away from the microscope. This one right here is called the coarse focus knob. This one over here that's a little bit more narrower is referred to as the fine focus knob, all right? And I'm going to tell you how to basically use these in a couple of minutes, all right? And then finally, pull this up. This right here, this whole thing right here is called the base, all right? The whole thing is called the base, all right? So again, make sure you know the portions of the microscope, because on your first laboratory practical, all right, that's what I'm going to test you on. Do you know this portion, all right? Do you know this portion, all right? Do you know what this portion is? Or do you know what this portion is, all right? So again, make sure you know the parts of the microscope, all right? So the first thing I want to show you before we actually start our exercises, all right, is how to basically use or care for your microscope, all right? So first of all, anytime you pull your microscope out, all right, first thing you want to point out is that your objective lens should be under scanning, all right? So that red objective should be there. Second thing, you want to make sure that your coarse focus knob is turned all the way down so that your stage is furthest away from your objective, all right? So when you pull your microscope out and you basically see that it's all the way up here and it's under a different objective other than a scanning objective, whoever used it last did it wrong, all right? You're my students, all right? I don't want you to be messing up on anything. So again, once you're done with the microscope, make sure it's under the scanning objective, and then make sure that the stage is furthest away from the objective, all right? Now notice, sometimes you may get to your lab bench and you may open up your cabinet and the microscope's gone. Typically, if the microscope's gone, that basically means there's something wrong with it and we're trying to get it repaired, all right? So what you're going to have to do is maybe go to another lab station or an empty lab station and pull another microscope out. I always say microscopes are like babies, all right? You don't carry a baby with one hand, you carry a baby with two hands. And so what's going to happen is I'm going to show you how to carry a microscope, all right? So again, if you basically have to get a microscope from another lab station, all right, you're basically going to have one hand on the arm and the other hand supporting the base, all right? So this is actually how you carry a compound light microscope. Don't carry it like this because you don't carry a baby like this unless they pooed. But anyway, you carry it close to you, all right? And so again, this is how you carry a compound light microscope, all right? Next, plug it in. And I do want to make a point. A lot of times when we basically pull them out or we put them away, we actually basically take the power cord and we basically wrap it around it. You don't want to wrap it around too tight, but you don't want it to be too loose, all right? And notice what I'm doing, all right, is when I'm putting this away, all right, I'm actually twisting it below the stage, all right? And the reason why is because if you twist it above the stage and you come into contact with these objective lenses, it may basically bend the objective lens, and what you've done is you've ruined the microscope, all right? So again, don't do it too tight, don't do it too loose, and when you do it, make sure that the tip of the power cord, just do it maybe twice, all right, so that they're basically in, all right? So this is basically how you basically put a microscope away, or how you basically pull it out to basically use it. So again, what I'm going to do again is I'm going to plug this in one more time. There we go. I'm going to turn my light off, all right? So the first thing I want to show you, all right, is because you may be brand new at doing this, and I do want to make a point. I can tell you how to use a microscope, all right? But whether or not you're going to be able to use the microscope is a different story. When I was in my PhD school, about half the graduate students still do not know how to use a microscope. It's almost like taking an art class, all right? Painting class, and I can basically show you this is how we're going to paint trees, all right? And some of you will paint beautiful trees, and the rest of you, I'm thinking like, you call that a tree? It's you either get it or you don't, all right? So first of all, what I'm going to do is I'm going to pull a slide. It could be any slide. It could be through this lab. It could be through other labs. I'm going to pull a slide. Make sure again that I'm under the scanning objective. Make sure again that the stage is furthest away from your objective lens. First thing I'm going to do is I'm going to turn on my lights. All right, you can actually see that my light's on. Make sure that your specimen is actually shining through the light. So when you look at it, you can actually use your adjustment knobs right here, your back to forth or your right to left adjustment knobs. Do that, all right, so just to make sure that that light is coming through your specimen, all right? If you wear glasses, all right, just kind of remove them, all right? First thing I want to do is I want to stare through here. I may have to adjust my eyepieces to make sure. And again, you're going to see a little pointer, all right? That should be there, all right? So again, when we're looking at this and I'm actually testing you, I may actually point to a cell or point to a structure and say, hey, identify that structure or what process is that cell going through or whatever, all right? First thing I'm going to do is I'm going to use my coarse focus knob, all right? And basically turn it, all right, until everything comes into focus. Now, once it's in focus, all right, that looks pretty good for me, all right. Then I can basically adjust my adjustment knobs to make sure it's centered. Beautiful, all right, beautiful. And notice again, this is under the scanning objective. So for the coarse focus knob, this is the only time you should actually use this is when you're basically focusing under the scanning objective. Now, these microscopes are something referred to as parfocal. What does that mean? In other words, if you have it in focus under one objective, if I increase or decrease, in other words, if I change the objective lenses, it should still be in focus. That isn't always true, but here what I'm going to do is I'm going to go from the scanning objective to low power objective, all right? When I'm looking through here, I can still see it, but it's kind of out of focus. Now, if I grab my coarse focus knob and I twisted it, what's going to happen is my slide is going to come up and hit my objective. You're going to crack or break your slide. You're going to scratch your objective or both, all right? So under low power, scanning, or if you're using the oil immersion lens, do not twist this coarse focus knob. You only used your fine focus knob. So again, it's under, we're going from scanning to low power. What I'm going to do is look at it. I'm going to use my scanning. Brilliant. All right, brilliant. Same thing if I go to high power. And look how close the objectives are to the slide. So again, do not grab onto this coarse focus knob and twist it. You're going to ruin your slide, the microscope, or both, all right? And please don't do that. Please, please, please. All right. So again, this is basically how you view it. And for us, all right, at least when we start off, all right, once I basically take my microscope slide, put it in the back, and I'm going to grab another microscope slide, always make sure that the stage is furthest away from your objective and always go back down to the scanning objective and start over, all right? This will actually teach you how to use a microscope, all right? This will actually teach you how to use a microscope. Now, say you're looking through a microscope and either there's basically dust or dirt on either your ocular lens or your objective lens, all right? Well, a lot of people will want to lick it, all right? Use paper towels, all right? Don't do any of that, all right? The best way to do this is through something referred to as lens paper. And lens paper are in these little drawers, all right? They should be right above your microscope, the drawer's right above the microscope. What I want to do is I'm going to grab one of these things and take the lens paper out. You don't have to put any solvent or anything on this. As a matter of fact, I wouldn't put solvents on here, period, all right? What you're going to do is take it, and you don't have to put a lot of pressure, all right? Because if you put a lot of pressure, again, you're going to crack something, all right? Basically, take it And what I'm doing, let me twist this around, sorry. This is how I'm doing. I'm just putting light pressure and I'm basically getting all that dust and dirt off one ocular lens. Then I'm going to do the same thing, but I'm going to use a different piece of the lens paper and do it for the same thing. All right. You can also do it again for your objectives. All right. And if you do that, that'll make all the difference. Now, sometimes your microscope slide may have some dirt on it. All right. And here you don't actually have to use lens paper. You could actually use like a paper towel, all right? But again, don't put a lot of pressure on it. And again, don't put any sort of chemicals that actually maybe like dissolve the perm out that actually sticks the cover slip to the slide. Just wipe it off, all right? And that's it, all right? So again, use the lens paper, all right? Next thing, which I've seen a lot of people do, which I think I made that mistake earlier, is scooting the microscope, all right? When you scoot the microscope, all right, notice that the light, all right, basically it's going to travel in a single direction, all right? But notice that your ocular lens is basically off here. So again, the light, it's got to pass up through here and then pass up through here. So inside this body tube, there's a prism, all right, that's basically going to take the light from your specimen, all right, and basically shine it through your ocular lens. Once you start to scoot that microscope, it'll vibrate that microscope. And if that prism comes loose, all right, and I can actually tell by looking at it that the prism's loose because some parts is going to be in focus and the other part is going to be out of focus. And again, what you've done is the microscope needs to be repaired again, all right? So try not to scoot the microscope. Once you put your microscope in a certain position, let it sit, all right? If you do have to move it, all right, again, pick it up and then move it instead of scooting it, all right? So again, that's basically how we basically use the microscope, all right? I'm thinking what we can do is go ahead and start with our exercises, all right, that I've got for today, all right? So I've got 4 exercises that I want you to do today. So the first one we're going to do is actually something referred to as the letter E, all right? So again, I've got microscope slide box right here called the letter E, and what I'm going to do is open it and pull one of these letter E's out. And notice when you basically put this under your microscope, all right, again, starting off on your microscope, all right, again, the stage has to be furthest away from the objective, and you want to start off under the scanning objective, which is the red objective, all right? What you're going to do is look at your microscope and look at it like you were reading it, like either off a computer screen or something you wrote down, a magazine or whatever, all right? And I can actually do this. I don't know if you can actually see it. I don't know if you can actually see it. This is the letter E, and what I'm going to do is I can read it. It's right side up. It's everything. It's not upside down. What I'm going to do is I'm going to put it under my microscope, all right? Make sure that the light is actually shining up through the letter E. And again, if you have glasses, put them on top of your head, look through it, and what I'm going to do is basically take my coarse focus knob, twist it until it comes into focus, all right? And that's it, all right? Now notice when you look at it, it's going to be a little bit different. It's going to be upside down and backwards, all right? So that's the whole purpose, is when you're looking at it through the microscope, you're not actually seeing the object itself, you're actually seeing the image of the object, all right? And notice again, there's 2 questions I have you. If I say move the microscope stage to the left, all right, what's going to happen is your letter E is actually going to move to the right. If I say move it to the right, the letter E is actually going to move to the left, all right? So everything's flip-flopped, all right? And that's what I'm saying. These mechanical stages actually work well. Supposing if you don't have the mechanical stages and you have to use your fingers, you have to be aware on what you're looking at, all right? Now note, we're only going to look at the letter E under the scanning objective, all right? If you go to like low power, it's so big, it's going to be hard to focus in anyway, all right? That's basically showing you that when you look at an object, what you're doing is you're seeing an image that's upside down and reversed, all right? Upside down and reversed. Now the next exercise I want you to look at is this one right here. This is something as exercise B. This is something referred to as the colored threads. Again, make sure the letter E is put back in the slide box, and when I pull this out, these are the colored threads. There's actually 3 colored threads out here, so it's blue, red, and yellow, all right? And again, make sure that your stage is furthest away from your objective, all right? Make sure you're under the scanning objective. Again, put it under the clips, all right, the microscope clips, all right? Make sure that you can actually see the light passing up through all three of the colored threads. And what I'm going to do again is use my coarse focus knob, and I'm going to twist it until everything gets into focus, all right? Make sure that when you do this, that you can basically adjust it, all right, using either the back, forth, or right to left adjustment knobs. You may have to adjust it using your fine focus, all right, but you want to make sure that each thread, regardless of whether or not it's blue, red, or yellow, all right, is in focus, all right? So once it's in focus, all right, I'm going to ask you, all right, are all three threads, all three colored threads in focus, at least under the scanning? It should be yes, all right? But now what I'm going to do, all right, is I'm going to say, switch the magnification or increase the magnification. And what you're going to do is you're going to go from the scanning objective to the low power objective. And again, because they're parfocal, you may have to adjust your adjustment knob just to make sure you can see all three of the colored threads. But again, you don't want to grab this coarse focus knob. You just want to use your fine focus knob and again, try to focus, all right? And when I'm focusing here, all right, usually I can see two threads that are in focus, but the other one is going to be out of focus, all right? Which is interesting. So the whole purpose of this exercise is that once you increase your magnification, the focusable field that you're actually looking at is going to get smaller and smaller and smaller. Now what I'm going to do is I'm going to say, hey, go from your low power objective to the high power objective. And again, when I'm looking at this, I'm going to basically use it. And keep in mind, when you go to the high power, it's going to be hard to see all three threads because, again, it's going to be magnified. And what's going to happen is I'm only seeing two threads right now. And I can actually focus on one, more or less, but the other one that's underneath it is extremely out of focus. And again, I'm only using my fine focus, not my coarse focus, all right? So again, once you increase your magnification to low power, high power, or the oil immersion, only use your fine focus, all right? And so again, increase in magnification, you're basically decreasing the field of focusable view that you can actually see. And notice how close this objective is to my specimen, all right? So again, don't use your coarse focus. And when you're done, all right, what I would do is go back to your scanning, twist it, and then put this away, all right? So again, we're basically done with the colored threads. Now, the next two laboratory exercises, all right, what we're going to be doing is making slides ourselves. Now, most of what you're going to see, all right, throughout the semester is preserved and stained. What does that mean? In other words, the specimen's killed, it has to be preserved, some of it has to be sliced, and then it's going to have to be stained with specific dyes, all right? Well, what we're going to be doing is actually looking at something that's sort of living, all right, although it's going to be dead afterwards. We're going to be looking at onion epidermal cells, all right? So we're going to be looking at cells from plants. We're also going to be looking at animal cells. They're basically going to be your cells, all right? So the first thing we're going to do is basically look at onion epidermal cells, all right? So what I want to do is I want to show you how this is done. I'm going to pull this back and put this over. What I've done is I've basically grabbed either like a yellow onion, a white onion, or a purple onion. Sometimes they call it red onion. And I basically cut the leaves, all right, around the bulb, all right, up in here. And usually what you do is you let them soak for a couple of hours, all right? What's going to happen is you're going to grab a clean microscope slide. Sometimes they're in nice little boxes, all right? But if we actually have some microscope slide that some students have already used, we can actually use those, all right? Because we use these clean microscope slides over and over again. What's going to happen is I'm going to basically take this clean microscope slide and I'm just going to put just a little drop of water. It's just a little drop of water. Not a lot, all right? But it's a little drop of water. Next, what I'm going to do is grab some forceps, all right? Some people call these tweezers, whatever you want to do, forceps, tweezers. And what I'm going to do is grab one of these things out, all right? And kind of use my fingers, all right? What you want to do is basically use these tweezers, all right, to basically grab these epidermal cells. They kind of look like it's like a thin membrane. You can kind of see it right there. See this thing right here? This thing right here? These are the epidermal cells. You don't need the entire thing, although if you want to, go ahead and do it. So I'm going to grab this, and what I'm going to do is I'm going to use my forceps and I'm actually going to stick it on the slide. And you kind of want it to be folded, basically be flat on the slide, not folded over, because if it's folded over, it's going to be hard to actually view the plant cells, all right? But notice again, when we basically put living cells on a light microscope, all right, the light basically shines up from them and they're translucent, so they're going to be almost impossible to see. What we're going to do is we're going to take some iodine, all right, and keep in mind if you're wearing nice clothes, all right, and I'm not really worried about my clothes, but if you're wearing nice clothes and you don't want it to stain, you might want to have an apron, and if you don't want to get like iodine or methylene blue on your fingers, you might want to wear gloves, all right? But what we're going to do is I'm just going to add, again, I'm not going to douse it, all right? Just a small drop of iodine, and that's it, all right? Then what I'm going to do is grab these things right here. They're called cover slips or cover glasses, all right? And be careful with these, all right? Because regardless of whether or not they're plastic or glass, if you break them, they can scratch you, all right? They can basically cut you open. And so what I'm going to do again is take one of these out, all right? And basically stick the cover glass or cover slip over here. And what I'm going to do is I'm going to have something that looks like this, all right? What I'm going to do is stick it under my microscope, all right, and view it. Usually for this one, all right, you're probably going to view it under low power, all right, for these cells, because plant cells tend to be just a little bit larger than animal cells, all right? What you can actually see when you actually look through this, all right, you can actually see the cell wall and you can actually see the nucleus. Pretty much the other structures you're probably not going to be able to see. But one thing I want to point out, when we basically talk about resolution, all right, it's basically when two smaller objects are close together, can we actually see them as two objects or do we see them as one object? Hopefully you know that each plant cell is surrounded by something referred to as the primary cell wall, all right? What you're going to be doing is looking at a couple of plant cells that are all next to each other, all right? When you look at this, you only see one plant cell wall, which basically means that the primary cell wall of plant cells is less than 0.2 microns. And so instead of seeing two plant cell walls, you only see one. And this basically is the limitation of the resolution for a compound light microscope. If I was using an electron microscope, using an electron beam, I could actually see the two cell walls. I could actually see that little middle layer, which is referred to as the middle lamella, all right? So resolution just for light microscopes is kind of limited, all right? So again, that's what you're looking at. Now, once you're done with this, all right, what you're going to do is you're going to clean everything up, all right? You're not going to make a mess and just leave it for me to clean up, all right? So again, take your slide, all right, and basically wash everything off, all right? Wash everything off at one of the sinks, all right? What you're going to do is you don't really have to use soap or water unless it's extremely dirty. Wash the iodine, wash the epidermal cells, the onion epidermal cells off, and clean it up, and basically put it on paper towels to let it dry, because we can actually use the microscope slide and the cover glass or the cover slip over again, all right? Now, this last experiment that we're going to do, all right, are something referred to as cheek cells, all right, human cheek cells, all right? So again, what you're going to do again is grab a clean microscope slide. Again, what you're going to do is put just a small drop of water on here, all right, just a small drop, all right? If it's too big, it's going to mess everything up, all right? Now what you're going to do is basically take a flattened toothpick, all right, a flattened toothpick, all right? And these are cheek cells, so these are basically going to be epidermal cells that basically lie within your oral cavity. And what you're going to do is you're actually going to scrape the inside. You don't want to put too much pressure on it because you don't want to like injure yourself or scratch the inside of your mouth, all right? Do like that. And then what you're going to do is you're basically going to put that in that water, all right? Just kind of push it around to basically spread all those cells out. Now, once you're done with the toothpick actually goes in the trash, all right? So again, make sure you throw your toothpicks away, all right? Then what you're going to do is take this dye right here, and again, if you don't want your clothes to be stained, wear an apron, all right? And this will also stain, just like iodine, it will also stain your skin too. So if you don't want to basically stain it, you can basically wear gloves. I'm not wearing gloves because I think I know what I'm doing. I've done this for years. But what you're going to do, again, is put a small drop of methylene blue. Not a big drop, but a small drop, and that's it. Then what you're going to do is come over here again, put that cover slide or cover slip, all right, or on your microscope slide. And then what you're going to do is you're going to have something that looks like this, all right? So again, this is the slide, the prepared slide I've done. What you're going to do again is put it under the microscope. Again, make sure you're under scanning objective, all right? Focus it, and probably what you were going to do is you're actually going to view it under low power. So low power, you can actually see the cells, all right? And notice again, these are animal cells. So there's going to be no cell wall. You can basically see kind of like the outline of the cell, which is like the plasma membrane, and you can actually see the nucleus, all right? Now you may be able to go up to high power, all right, to see it. Although when you go to high power, because again, these things are three-dimensional, all right, you're going to lose some of your focus, all right? But again, you could actually view it these under low power, all right? You can actually see, and these are your cells, all right? These are your cheek cells, all right? Which again, they're epidermal cells, and so they basically give rise to every about 5 to 6 days, you basically replace these things over and over again, all right? And so that's what you're doing. Once you're done again, all right, make sure you basically clean it up, go over to the sink, all right, wash everything off, all right, make sure that there's no cells on here, no methylene blue, wash everything off. Make sure again that we can actually use the microscope slide and the cover slips or the cover glasses again, all right, and then put these on basically a piece of paper towel to let them dry out, all right? Now, when you're done, all right, make sure your microscope is off. Make sure you're under your scanning objective. Make sure that your stage is furthest away from your objective. Again, what you're going to do is you're going to wrap the power cord, not too tightly or too loosely, around underneath the stage, all right? Put the protective covering back on and then basically stick it back where you basically found it, all right? So again, this is basically the introduction to how to basically use the compound light microscope. In the next lab, all right, lab 2, we're actually going to be using the compound light microscope to actually view a process referred to as mitosis and cytokinesis in both plant cells and animal cells. I'll see you then.